The oceans cover approximately 71% of Earth's surface, containing roughly 97% of all water on the planet, and this simple quantitative fact carries implications that extend far beyond geography into the fundamental processes that have shaped terrestrial existence for billions of years. The total volume of ocean water measures approximately 1.335 billion cubic kilometres, a figure that represents such an enormous mass that the water itself exerts measurable gravitational effects on surrounding continental landmasses and influences the distribution of weight across the planetary surface in ways that affect Earth's rotation. The average depth of the global ocean reaches approximately 388 metres, though this mean value obscures tremendous variations ranging from shallow coastal shelves extending only tens of metres below the surface to abyssal trenches plunging beyond 11 kilometres into the lithosphere. The deepest known point, located in the Mariana Trench's Challenger Deep, reaches approximately 10,135 metres below sea level, a measurement that was first established with any precision only in 1960, when the bathyscaphe Trieste carried Jacques Picard and Don Walsh to the seafloor in a descent that took nearly 5 hours and revealed pressures exceeding 1,000 atmospheres. Understanding how human knowledge of ocean depth developed requires examining the technological and institutional frameworks that made such measurements possible, because for most of recorded history, the actual dimensions of the marine realm remained entirely unknown to coastal populations who had developed sophisticated practical knowledge of surface waters without any capacity to investigate what lay beneath. Ancient Mediterranean civilizations operated extensive maritime trading networks connecting Egypt, Phoenicia, Greece, and the Western Mediterranean basin, yet their conceptual frameworks for understanding the sea emphasize surface phenomena, wind patterns, seasonal navigability, and the locations of dangerous shoals rather than any systematic investigation of vertical dimensions. The Greeks developed philosophical speculations about the nature of the sea, with thinkers such as Aristotle observing and cataloguing marine organisms with remarkable accuracy, but the technical means to measure ocean depths beyond the reach of weighted lines simply did not exist. Sounding with lead weights attached to marked ropes could establish depths in harbours and nearshore waters, and navigators used this technique extensively for safe passage through unfamiliar coastal areas. But the practical limits of such measurements extended only to a few hundred metres at most, leaving the vast majority of the ocean floor entirely inaccessible to quantitative investigation. The development of more systematic approaches to ocean measurement emerged gradually through the early modern period, driven partly by commercial and naval interests that required better charts, and partly by the growing influence of empirical natural philosophy that emphasized measurement and observation as foundations for understanding the physical world. By the 17th century, natural philosophers associated with institutions such as the Royal Society of London were beginning to formulate questions about ocean properties that demanded new investigative techniques, including attempts to measure not only depth, but also temperature, density, and the characteristics of water at various levels. Robert Boyle conducted experiments on the compressibility of seawater and speculated about conditions at great depths, recognizing that pressures would increase substantially with descent, and wondering what effects this might have on living organisms and physical processes. These early investigations remained limited by available technology, but they established intellectual frameworks that would guide more systematic research in subsequent centuries, framing the ocean as a domain of measurable physical properties rather than simply a surface upon which ships travelled and from which fish were harvested. The 19th century brought transformative developments in ocean science, driven by a combination of technological innovation, imperial expansion, and the institutionalization of scientific research within governmental and academic structures. The laying of transatlantic telegraph cables beginning in the 1850s required unprecedented knowledge of seafloor topography, creating commercial incentives for detailed bathymetric surveys that revealed the Atlantic's basic structure for the first time. These cable-laying expeditions discovered that the ocean floor was not a uniform plane but contained substantial topographic variation, including a prominent rise running through the central Atlantic that would eventually be understood as part of the global mid-ocean ridge system. The cable companies employed surveying techniques that combined improved sounding equipment, with systematic approaches to data collection, generating charts that represented a quantum leap in human understanding of submarine geography. Simultaneously, the development of oceanography as a formal scientific discipline proceeded through expeditions such as the famous Challenger expedition of 1872-1876, which circumnavigated the globe while conducting systematic measurements of temperature, salinity, currents, seafloor sediments, and biological specimens at stations distributed across all major ocean basins. The Challenger expedition deserves particular attention because it established methodological standards and generated baseline data that remained foundational to oceanographic research for decades afterward, while also revealing the institutional complexities of organising large-scale scientific investigations during the Victorian era. The expedition was funded by the British government and organised through collaboration between the Royal Society and the Royal Navy, reflecting the close relationships between scientific institutions and imperial state apparatus that characterised much 19th century research. The ship carried a scientific staff led by Charles Wyville Thompson. Along with naval officers and crew conducted the daily operations of sailing, while also participating in the collection of samples and measurements. Over the course of nearly 4 years, the expedition established 362 observation stations where comprehensive measurements were taken, recovering samples of water, sediment and organisms that filled hundreds of containers and would take 2 decades to analyse fully. The published reports eventually extended to 50 volumes containing descriptions of thousands of previously unknown species and establishing fundamental facts about ocean chemistry, temperature distribution, and sediment composition that transformed understanding of the marine environment. What the Challenger expedition and its successors revealed was an ocean far more complex and dynamic than previous generations had imagined, characterized by intricate patterns of circulation, pronounced vertical stratification, and biological communities adapted to conditions ranging from sunlit surface waters to the perpetual darkness and crushing pressures of abyssal depths. The discovery that the deep ocean contained abundant life overturned earlier assumptions that had extrapolated from the known absence of photosynthesis below the euphotic zone to conclude that deep waters must be essentially lifeless. Victorian scientists had debated whether life could exist at great depths, with some arguing that pressure, cold, and darkness would preclude biological activity, while others pointed to organisms recovered in fishing nets and dredges as evidence that life penetrated to considerable depths. The Challenger's systematic sampling definitively settled this question by recovering diverse fauna from all depths reached, including previously unknown species of fish, invertebrates, and microorganisms that demonstrated successful adaptation to deep-sea conditions. This discovery opened new questions about how energy and nutrients reach these deep communities, questions that would not be fully answered until the 20th century brought understanding of the biological pump and marine snow that transfer organic matter from productive surface waters to the deep ocean. The temperature structure of the ocean proved to be one of the most consequential discoveries of early oceanographic research, revealing a fundamental layering that influences everything from biological productivity to global climate. Surface waters in tropical and temperate regions receive solar heating that maintains temperatures ranging from approximately 25 degrees Celsius in tropical waters to 10 degrees or lower in higher latitudes. But below the mixed surface layer, temperatures decline rapidly through a zone called the thermocline, before reaching the cold, dense waters that fill the deep ocean at temperatures typically between 0 and 4 degrees Celsius. This thermal stratification creates barriers to vertical mixing that have profound implications for nutrient distribution and biological productivity, because while surface waters receive abundant sunlight for photosynthesis, they become depleted of essential nutrients that sink with organic matter and accumulate in deeper layers. The most productive regions of the ocean occur where physical processes break down this stratification, bringing nutrient-rich deep waters to the surface through upwelling driven by winds, topography, or density-driven circulation patterns. Coastal upwelling zones along the western margins of continents, particularly off Peru, California, Northwest Africa, and Namibia, support some of the world's most productive fisheries precisely because prevailing winds and the Coriolis effect drives surface waters offshore, drawing cold, nutrient-rich water upward from depth. The relationship between ocean circulation and global climate represents one of the most important areas where oceanographic research intersects with broader questions of planetary function and human welfare. The ocean's enormous thermal capacity allows it to absorb, store, and transport vast quantities of heat, moderating temperature extremes that would otherwise characterize continental interiors and redistributing energy from equatorial regions toward the poles. The Gulf Stream system, to take a particularly well-studied example, carries approximately 1.4 petawatts of heat northward through the Atlantic, warming northwestern Europe by an estimated 4 to 6 degrees Celsius, compared to what would be expected from latitude alone. This single current system transports a volume of water exceeding 100 times the combined flow of all terrestrial rivers, yet it represents only one component of a global circulation pattern that connects all ocean basins through what oceanographers term the thermohaline circulation or global conveyor belt. The driving forces behind this circulation involve the density differences created by temperature and salinity variations, with cold, salty water forming in high-latitude regions and sinking to fill the deep ocean basins before slowly spreading toward the equator over timescales measured in centuries. Understanding this circulation pattern and its sensitivity to changes in freshwater input, temperature, and other variables has become critically important for projecting future climate trajectories under conditions of increasing atmospheric carbon dioxide concentrations. The chemistry of seawater provides another domain where detailed knowledge has accumulated gradually through centuries of investigation, revealing properties that influence everything from the color of the ocean to its capacity to support life and regulate atmospheric composition. The average salinity of seawater measures approximately 35 parts per thousand, meaning that each kilogram of seawater contains about 35 grams of dissolved salts, predominantly sodium chloride, but including significant quantities of magnesium, sulfate, calcium, potassium, and bicarbonate ions. This composition remains remarkably constant across different ocean regions, despite substantial variations in the processes that add or remove water, suggesting that the ocean has achieved a kind of chemical steady state over geological timescales through the balance between inputs from rivers and hydrothermal vents against removal by biological activity, chemical precipitation, and interaction with the seafloor. The constancy of major ion ratios was recognized by 19th century chemists and formalized in what became known as the principle of constant proportions, which holds that while total salinity varies from place to place depending on precipitation, evaporation, and mixing, the relative abundances of major ions remain fixed. This principle has proved essential for oceanographic research because it allows salinity to be calculated from measurements of any single major ion, simplifying the analytical procedures required for water mass identification and circulation studies. Beyond the major ions that define seawater's basic chemistry, the ocean contains trace quantities of virtually every element in the periodic table, including metals, gases, and organic compounds that play essential roles in biological processes despite their minute concentrations. Iron, for example, occurs in open ocean surface waters at concentrations typically below 1 nanomolar, yet this element serves as an essential micronutrient for phytoplankton and appears to limit biological productivity across vast regions of the Southern Ocean, subarctic Pacific, and equatorial Pacific, where major nutrients such as nitrogen and phosphorus remain underutilized. The recognition of iron limitation emerged relatively recently in oceanographic history, with John Martin and colleagues conducting pivotal experiments in the late 1980s and 1990s that demonstrated dramatic increases in phytoplankton growth following artificial iron additions to surface waters. These findings generated considerable interest in iron fertilization as a potential mechanism for enhancing ocean carbon uptake and mitigating climate change, though subsequent research has revealed substantial complications, including the uncertainty of carbon export to deep waters and concerns about unintended ecological consequences. The iron limitation story illustrates how understanding of ocean biogeochemistry continues to develop through the interaction of field observations, laboratory experiments, and theoretical models, With new discoveries regularly revising established views of marine processes, the biological productivity of the ocean ultimately depends on photosynthesis conducted by phytoplankton, a diverse assemblage of microscopic organisms including diatoms, dinoflagellates, coccolithophores, and cyanobacteria that drift in sunlit surface waters and convert dissolved carbon dioxide into organic matter using solar energy. These organisms, though individually invisible to the naked eye, collectively produce approximately half of all oxygen generated through photosynthesis on Earth and form the base of marine food webs that support everything from zooplankton to whales. The distribution of phytoplankton productivity varies enormously across ocean regions and through time, with the highest values occurring in coastal upwelling zones, nutrient-enriched polar waters, and areas where physical mixing brings deep nutrients to the surface. Satellite remote sensing has revolutionized understanding of phytoplankton distribution by enabling global observation of ocean color, which reflects the concentration of chlorophyll and other photosynthetic pigments in surface waters. Instruments such as the Seaviewing Wide Field of View sensor, which operated from 1997 to 2010, and its successors have produced continuous records of ocean biological activity that reveal seasonal cycles, interannual variations, and long-term trends in marine productivity. The fate of organic matter produced by phytoplankton determines the ocean's role in the global carbon cycle, a system of such importance that understanding its functioning has become central to climate science and environmental policy. When phytoplankton die, or are consumed by zooplankton, a fraction of the organic matter they contain sinks toward the seafloor as particles collectively termed marine snow, which includes fecal pellets, dead cells, and aggregates formed through physical and biological processes. This sinking organic matter carries carbon from the surface ocean, where it was fixed through photosynthesis, into the deep ocean, where it is either remineralized by bacteria and returned to dissolved form, or reaches the seafloor and becomes incorporated into sediments. The efficiency of this biological pump in transferring carbon to depth depends on factors including the size and sinking rate of particles, the intensity of bacterial degradation during descent, and the depth at which most remineralization occurs. Oceanographers estimate that the biological pump sequesters approximately 2 to 3 billion tons of carbon per year in the deep ocean, a flux that significantly exceeds anthropogenic emissions and that helps maintain atmospheric carbon dioxide concentrations lower than they would otherwise be. The ocean's role as a carbon reservoir extends beyond the biological pump to include physical and chemical processes that dissolve atmospheric carbon dioxide directly into surface waters and transport it to depth through circulation. Carbon dioxide is more soluble in cold water than in warm water, so polar regions where surface waters cool before sinking serve as particularly effective sinks for atmospheric carbon. The dissolved carbon dioxide undergoes chemical reactions with water and existing carbonate and bicarbonate ions, establishing equilibria that buffer the ocean's pH and determine its capacity to absorb additional carbon from the atmosphere. This buffering capacity is not unlimited, however, and the absorption of anthropogenic carbon dioxide emissions is gradually shifting the equilibrium toward higher hydrogen ion concentrations, a process termed ocean acidification that has measurable effects on the saturation state of calcium carbonate minerals essential for shell-building organisms. Since the Industrial Revolution, the average pH of surface ocean waters has declined by approximately 0.1 units, representing a roughly 26% increase in hydrogen ion concentration, and projections suggest further declines of 0.3 to 0.4 units by 2100 under high emission scenarios. The implications of ocean acidification for marine ecosystems remain subjects of active research, with laboratory experiments, field observations, and modelling studies providing complementary perspectives on how organisms and communities may respond to changing water chemistry. Organisms that build shells or skeletons from calcium carbonate, including corals, mollusks, echinoderms, and many planktonic species, face particular challenges as declining saturation states make shell formation more energetically costly and increase dissolution rates for existing structures. Coral reefs, which support extraordinarily diverse communities despite covering less than 1% of the ocean floor, appear especially vulnerable to the combined stresses of warming waters, which cause bleaching events when temperatures exceed tolerable ranges, and acidification, which impairs the calcification processes essential for reef growth. Field studies at naturally acidified sites such as volcanic carbon dioxide seeps in Papua New Guinea and the Mediterranean have documented shifts in community composition towards species more tolerant of low pH conditions, suggesting that acidification may fundamentally restructure marine ecosystems rather than simply reducing productivity. However, the capacity of marine organisms to adapt to changing conditions through phenotypic plasticity or evolutionary change remains uncertain, and predictions about ecological futures necessarily incorporate substantial uncertainty about biological responses to novel environmental conditions. Turning from chemical properties to physical forces, the movements of ocean water encompass phenomena ranging from small-scale turbulence to planetary-scale circulation patterns, each operating on characteristic timescales and interacting with others in ways that determine how the ocean transports heat, nutrients, and organisms across vast distances. Surface currents driven by prevailing winds organize into large gyres that rotate clockwise in the northern hemisphere and counterclockwise in the southern hemisphere, reflecting the Coriolis effect's influence on moving fluids. These subtropical gyres accumulate warm surface water in their centres, creating regions of relatively low biological productivity, sometimes described as oceanic deserts, where nutrients are scarce and primary production limited. At the boundaries of these gyres, particularly along western margins where currents intensify into narrow swift flows such as the Gulf Stream, Kuroshio, and Agulhas currents, complex physical processes create fronts, eddies, and meanders that influence both local and downstream conditions. The study of ocean eddies has advanced considerably since satellite altimetry enabled routine observation of sea surface height variations associated with these rotating water masses, revealing that eddies transport significant quantities of heat, salt, and nutrients across ocean basins and play important roles in the overall energy budget of ocean circulation. Below the wind-driven surface layer, the deep ocean circulates according to different dynamics dominated by density differences created through heating, cooling, and freshwater fluxes at the sea surface. The formation of dense water masses capable of sinking to great depths occurs primarily in high-latitude regions where surface waters become cold and in some cases salty through ice formation that leaves salt behind in the remaining water. The North Atlantic deep water, one of the best studied deep water masses, forms primarily in the Nordic Seas and Labrador Sea when winter cooling increases density sufficiently for surface water to sink through the lighter waters below and spread southward along the western Atlantic basin at depths between roughly 1,500 and 4,000 meters. Antarctic bottom water, the densest water mass in the global ocean, forms around the margins of Antarctica, where extremely cold conditions and sea ice formation create water that sinks to the seafloor and spreads northward, eventually reaching as far as the North Atlantic and North Pacific. These deep water masses move slowly, requiring centuries to transit ocean basins, but they carry with them the temperature, salinity, and chemical signatures acquired at their formation sites, allowing oceanographers to trace their origins and pathways through measurements of water properties at distant locations. The timescales of deep ocean circulation have important implications for understanding both natural climate variability and the ocean's response to anthropogenic perturbations, because changes in deep water formation can propagate through the system only slowly, while surface conditions may respond much more rapidly. Paleoceanographic studies using sediment cores and other archives have documented past changes in ocean circulation that accompanied major climate transitions, including evidence that deep water formation in the North Atlantic weakened substantially during cold periods and strengthened during warmings. These circulation changes appear to have amplified climate variability by altering the distribution of heat between the hemispheres and affecting the ocean's capacity to sequester carbon dioxide from the atmosphere. Contemporary concerns about circulation changes focus partly on the possibility that increasing freshwater input to the North Atlantic from melting ice sheets and increased precipitation could reduce the density of surface waters and weaken deep water formation, potentially triggering a slowdown or reorganization of the thermohaline circulation with consequences for regional and global climate. Observational evidence for such changes remains limited, though some studies have suggested a weakening of the Atlantic meridional overturning circulation over recent decades, and climate models project continued weakening under future warming scenarios. The geological framework within which ocean circulation occurs has itself been transformed by scientific discoveries that fundamentally revised understanding of Earth's structure and dynamics during the 20th century. The recognition that the seafloor is not simply a passive basin, but an active geological domain where new crust forms and old crust is destroyed, emerged through the plate tectonics revolution of the 1960s, synthesizing observations from diverse fields including marine geology, seismology, and paleomagnetism into a unified theory of Earth's behavior. Mid-ocean ridges, which form a continuous mountain chain extending approximately 65,000 kilometers through all major ocean basins, represent sites where mantle material rises to fill the gap created as tectonic plates diverge, creating new oceanic crust that spreads laterally away from the ridge axis, at rates typically ranging from 1 to 10 centimeters per year. The youthful crust near ridge axes contrasts with a progressively older crust toward continental margins, a pattern documented through dating of magnetic anomalies preserved in seafloor rocks that record reversals in Earth's magnetic field at known times. At subduction zones, typically marked by deep ocean trenches, old oceanic crust descends back into the mantle, completing a cycle of creation and destruction that continuously renews the seafloor while recycling water, carbon, and other volatile elements between Earth's surface and interior. The implications of plate tectonics for ocean evolution extend beyond the production of new seafloor to include the opening and closing of ocean basins, the creation and destruction of connections between ocean regions, and the repositioning of continents in ways that fundamentally alter global circulation patterns. The Atlantic Ocean, for example, has been widening since the supercontinent. Pangaea began to break apart approximately 200 million years ago, with the spreading mid-Atlantic ridge creating new crust at rates of about 2.5 centimeters per year and pushing the Americas away from Europe and Africa. The Pacific Ocean, by contrast, has been shrinking as subduction consumes crust around its margins faster than spreading creates new crust at the East Pacific Rise, and paleographic reconstructions suggest that a much larger Pacific once existed during earlier geological periods. The opening of the Southern Ocean passages that allowed circumpolar circulation to develop, particularly the Drake Passage between South America and Antarctica and the Tasmanian Gateway south of Australia, appears to have been a crucial factor in Antarctic glaciation by thermally isolating the continent and enabling the development of the cold conditions necessary for ice sheet growth. These examples illustrate how ocean geography and circulation have evolved continuously through Earth's history, with present conditions representing just one frame in an ongoing transformation driven by the slow but persistent movements of tectonic plates. Hydrothermal vents discovered at mid-ocean ridges in 1977 represent one of the most remarkable findings of late 20th century ocean science, revealing ecosystems that derive their energy not from sunlight, but from chemical reactions involving reduced compounds released by volcanic processes. The initial discovery along the Galapagos Rift documented communities of large organisms, including giant tube worms, clams, and mussels, clustered around vent openings, where superheated water laden with dissolved metals and sulfides emerged from the seafloor. Subsequent exploration found similar communities at hydrothermal sites throughout the global ocean, each hosting assemblages of organisms adapted to the unusual chemical and physical conditions present at vents. The primary producers in these ecosystems are chemosynthetic bacteria and archaea that oxidize reduced compounds, particularly hydrogen sulfide, to obtain energy for carbon fixation, forming the base of food webs that support diverse higher organisms. Many vent animals harbour symbiotic chemosynthetic microbes within specialized tissues, obtaining nutrition directly from these internal partners rather than through conventional feeding, an arrangement that has evolved independently in multiple lineages and represents a fundamental departure from the photosynthesis-based energy capture that dominates surface ecosystems. The discovery of hydrothermal vent ecosystems prompted substantial revision of ideas about the limits of life on Earth and the potential for life on other planetary bodies, because it demonstrated that complex biological communities could thrive in the complete absence of sunlight given appropriate chemical energy sources. Theoretical work on the origins of life has increasingly focused on hydrothermal environments as potential sites for the emergence of the first living systems, with researchers noting that the chemical gradients, mineral surfaces, and steady energy supplies at vents provide conditions favourable for prebiotic chemistry and early metabolic pathways. Whether life actually originated at hydrothermal vents, at surface environments exposed to solar radiation, or in some other setting remains unresolved. But the existence of vent ecosystems demonstrates that photosynthesis is not a prerequisite for supporting diverse productive biological communities. This recognition has implications for astrobiology, because planetary bodies such as Jupiter's moon Europa and Saturn's moon Enceladus appear to possess subsurface oceans that may experience hydrothermal activity at their seafloors, potentially creating habitable environments despite their great distance from the Sun and the absence of any photosynthetic energy source. Cold seeps represent another category of chemosynthetic ecosystem, found where methane, hydrogen, sulfide, and other reduced compounds leak from seafloor sediments through geological processes distinct from the volcanic activity that drives hydrothermal vents. These seeps occur in diverse geological settings, including continental margins, where organic-rich sediments release gases through microbial decomposition, subduction zones where sediment compression forces fluids upward, and areas where underlying hydrocarbon reservoirs leak toward the surface. The communities at cold seeps share some characteristics with hydrothermal vent fauna, including dependence on chemosynthetic primary production and the presence of symbiotic bacteria in many dominant organisms, but they also include distinctive elements adapted to the lower temperatures and different chemical conditions typical of seep environments. Bacterial mats, tube worm aggregations, mussel beds, and methane-derived carbonate structures characterize many cold seeps, creating localized patches of high biomass and diversity within otherwise relatively unproductive deep-sea habitats. The recognition that cold seeps occur widely along continental margins has expanded understanding of chemosynthetic ecosystems beyond the isolated ridge environments where they were first discovered, revealing a global distribution of non-photosynthetic primary production in the deep ocean. The sediments that blanket the ocean floor provide archives of Earth's history extending back millions to hundreds of millions of years, preserving information about past climate, ocean chemistry, biological evolution, and geological events that can be accessed through scientific drilling and coring programs. Deep-sea sediments accumulate slowly, typically at rates of 1 to 10 centimeters per thousand years, building up sequences that record changing conditions through variations in composition, fossil content, and geochemical signatures. Micropaleontology, the study of microscopic fossils preserved in sediments, has proved particularly valuable for reconstructing past environments because the shells of foraminifera, diatoms, radiolarians, and other planktonic organisms reflect the temperature, chemistry, and productivity of the waters in which they lived. The oxygen isotope ratios in foraminiferal shells vary with both temperature and the isotopic composition of seawater, which itself changes as ice sheets grow or shrink, enabling the construction of detailed records of ice age cycles that have characterized the past few million years. These records have been essential for understanding the astronomical forcing of climate through variations in Earth's orbital parameters, a theory developed by Milutin Milankovitch in the early 20th century and confirmed by sediment core evidence beginning in the 1970s. The International Ocean Discovery Programme and its predecessor programs have coordinated scientific ocean drilling for more than 50 years, using specially equipped vessels to recover sediment and rock cores from sites throughout the global ocean. These programs have transformed understanding of Earth's history by providing continuous records spanning tens of millions of years at many sites, and by enabling direct sampling of oceanic crust and the organisms living within it. Among the significant discoveries enabled by ocean drilling are the documentation of the Paleocene-Eocene thermal maximum approximately 56 million years ago, when a massive release of carbon to the atmosphere drove global temperatures several degrees higher than present and caused substantial ocean acidification. The reconstruction of atmospheric carbon dioxide concentrations through geological time using various proxy indicators, and the discovery of a deep biosphere of microorganisms living within sediments and oceanic crust that may constitute a significant fraction of all living biomass on Earth. The deep biosphere findings have particularly broad implications because they suggest that life penetrates far deeper into Earth's crust than previously suspected, with metabolically active microbes detected at depths exceeding 2 kilometers below the seafloor in some locations. Returning to the upper reaches of the ocean, the interactions between atmosphere and ocean at the sea surface constitute a boundary zone where exchanges of heat, moisture, gases, and momentum drive both oceanic and atmospheric circulation. The ocean absorbs approximately 90% of the excess heat trapped by anthropogenic greenhouse gas emissions, a finding documented through systematic measurements of ocean temperature profiles extending back several decades and through more recent observations from the global Argo array of profiling floats. This heat absorption has profound consequences for both the ocean itself, which is warming throughout the water column, with corresponding effects on circulation, stratification, and biological communities, and for the climate system as a whole, which would otherwise experience more rapid atmospheric warming. The exchange of water between ocean and atmosphere through evaporation and precipitation drives the global hydrological cycle, with evaporation from the tropical oceans providing moisture that is transported by atmospheric circulation and precipitates over both oceanic and continental regions. The ocean's role in this cycle influences salinity distributions, which in turn affect density and circulation patterns, creating feedbacks between hydrological and oceanographic processes that operate on timescales from seasonal to millennial. Gas exchange between ocean and atmosphere extends beyond water vapor to include the climatically critical greenhouse gases carbon dioxide and methane, as well as oxygen, nitrogen, and various trace gases that influence atmospheric chemistry and air quality. The ocean's absorption of approximately 1/4 of annual anthropogenic carbon dioxide emissions represents a major component of the global carbon cycle. But this absorption comes at the cost of ocean acidification and represents a service that may diminish as surface waters approach saturation and as warming reduces the solubility of carbon dioxide. Methane emissions from the ocean derive primarily from anaerobic decomposition in sediments and from methane hydrate deposits that form under high-pressure, low-temperature conditions on continental margins and in Arctic regions. These hydrate deposits have attracted considerable research attention, both as potential energy resources and as potential sources of positive feedback to climate warming, because destabilization of hydrates through ocean warming could release large quantities of methane to the atmosphere, amplifying the initial warming signal. The actual magnitude of this feedback remains uncertain, with ongoing research attempting to constrain the sensitivity of hydrate stability to temperature changes and the pathways by which released methane might reach the atmosphere rather than being consumed by microorganisms in sediments or the water column. Marine ecosystems respond to the physical and chemical conditions set by ocean circulation, temperature distribution, and chemistry through ecological processes that determine the distribution and abundance of organisms from bacteria to whales. The biogeography of marine species reflects evolutionary history, dispersal capacity, and the environmental tolerances of different organisms, creating patterns of diversity and endemism that vary systematically across ocean regions. Tropical coral reef ecosystems support the highest marine biodiversity, with the Indo-Pacific Centre of Diversity harbouring particularly rich assemblages that decline toward the Atlantic and toward higher latitudes. The factors driving this diversity gradient remain debated, with hypotheses emphasising historical factors such as the greater age and stability of Indo-Pacific reef environments, ecological factors such as the availability of habitat and the intensity of competition and predation, and physical factors such as temperature ranges and the geometry of ocean basins. Understanding these patterns has practical significance for conservation planning because it helps identify regions of particular importance for protecting marine biodiversity and predicts how species distributions may shift as climate conditions change. The exploitation of marine biological resources has a history extending back to prehistoric times, with archaeological evidence documenting fishing and shellfish harvesting by coastal populations throughout the world for tens of thousands of years. The industrialization of fishing during the 19th and 20th centuries transformed marine exploitation from a largely local activity using simple technologies to a global industry employing sophisticated vessels, gear, and electronic equipment capable of locating and capturing fish with unprecedented efficiency. Global marine fish catch increased roughly fivefold between 1950 and the late 1990s, reaching approximately 80 million tons annually before levelling off and beginning to decline in some regions as overexploited stocks contracted. The scientific assessment of fish populations developed in parallel with industrial fishing, establishing concepts and methods for estimating stock size, sustainable yield, and the effects of fishing mortality that inform contemporary fisheries management. Maximum sustainable yield, a concept introduced in the mid-20th century, represents the largest catch that can theoretically be taken from a stock indefinitely without causing population decline, and this concept has been central to management frameworks despite recognised limitations and the practical difficulties of accurate stock assessment. The history of specific fisheries illustrates the dynamics of marine resource exploitation and the challenges of achieving sustainable management in the face of economic pressures, scientific uncertainty, and institutional constraints. The northern cod fishery off Newfoundland and Labrador provides a particularly instructive example, having supported substantial fishing activity for 5 centuries before collapsing in the early 1990s to a small fraction of historical abundance. European fishers exploited Grand Banks cod from the early 16th century onward, attracted by reports of enormous abundance from explorers such as John Cabot, and this fishery grew to become one of the most economically important in the North Atlantic. By the mid-20th century, factory trawlers from distant water fleets were taking catches far exceeding anything in previous history, and cod populations began to decline despite management measures that proved inadequate to constrain fishing effort. The 1992 moratorium on commercial cod fishing, imposed when scientific surveys revealed population collapse, had devastating economic and social consequences for Newfoundland communities that had depended on the fishery for generations, while the expected recovery has been slow and incomplete more than 3 decades later. Analysis of this and other fishery collapses has identified multiple contributing factors, including excessive fishing mortality, scientific underestimation of stock declines, political reluctance to impose unpopular restrictions, and ecosystem changes related to climate and predator-prey dynamics. The regulation of ocean activities involves complex jurisdictional frameworks that have evolved substantially since the mid-20th century, responding to technological changes that expanded human capacity to exploit marine resources and to increasing recognition of the need for international governance of shared and high seas areas. The United Nations Convention on the Law of the Sea, adopted in 1982 after nearly a decade of negotiation and entering into force in 1994, established the contemporary framework for maritime jurisdiction, defining coastal state rights over territorial seas, extending 12 nautical miles from shore, exclusive economic zones extending 200 nautical miles within which states have sovereign rights over resources, and continental shelf rights that can extend further where geological conditions permit. The Convention also established principles for the management of high seas areas beyond national jurisdiction, which cover approximately 64% of the ocean surface, and contain resources ranging from fish to minerals that have attracted increasing commercial interest. Subsequent agreements have addressed specific issues, including the conservation of straddling fish stocks that migrate across jurisdictional boundaries, the governance of deep-sea mining in the International Seabed Area, administered by the International Seabed Authority, and most recently a treaty on the conservation and sustainable use of marine biological diversity in areas beyond national jurisdiction, concluded in 2023. Deep-sea mining represents an emerging frontier of ocean resource exploitation with significant potential consequences for marine ecosystems that remain poorly understood and incompletely protected under existing governance arrangements. The mineral resources of interest include polymetallic nodules found on abyssal plains at depths typically between 4,000 and 6,000 meters, polymetallic sulfide deposits associated with hydrothermal vents, and cobalt-rich ferromanganese crusts forming on seamount slopes. These deposits contain metals including manganese, nickel, cobalt, copper, and rare earth elements that are increasingly in demand for renewable energy technologies and electronic devices, creating economic incentives for extraction despite the technical challenges and environmental risks involved. The International Seabed Authority has granted exploration contracts covering substantial areas of the Pacific, Atlantic, and Indian Ocean sea floors, and pressure to move from exploration toward commercial extraction has intensified as terrestrial deposits become depleted or face development constraints. Environmental concerns focus on the destruction of seafloor habitats during mining operations, the suspension of sediments that could affect organisms over large areas downstream of mining sites, and the potential loss of unique biological communities, particularly at hydrothermal vents where endemic species might be extirpated by extraction activities targeting sulfide deposits. Marine pollution takes many forms, ranging from the visible accumulation of plastic debris to invisible but consequential contamination by persistent organic pollutants, heavy metals, and excess nutrients. The recognition of plastic pollution as a major environmental issue has grown substantially since the early 2000s, driven partly by accumulating scientific evidence and partly by public attention to phenomena such as the accumulation of floating debris in the subtropical gyres, where convergent currents concentrate buoyant materials in semi-permanent garbage patches. The actual distribution of plastic debris in the ocean is more complex than popular accounts often suggest, with surface accumulations representing only a fraction of total marine plastic, while substantial quantities have been found in deep-sea sediments, coastal environments, and the tissues of marine organisms from the smallest zooplankton to the largest whales. Microplastics, defined as particles smaller than 5 millimeters, have attracted particular research attention because they are ingested by organisms throughout marine food webs and may transfer contaminants or cause physical harm, though the population-level consequences of microplastic exposure remain actively debated. The sources of marine plastic pollution include direct disposal of waste at sea, land-based runoff carrying debris from inadequate waste management systems, and the fragmentation of larger items into progressively smaller particles through weathering and mechanical breakdown. Nutrient pollution, primarily from agricultural runoff, and urban wastewater carrying nitrogen and phosphorus compounds, has created expanding zones of low oxygen or hypoxic conditions in coastal waters worldwide, where excess primary production leads to bacterial decomposition that consumes dissolved oxygen faster than it can be replenished. These hypoxic zones, sometimes termed dead zones in popular usage, can render substantial areas of seafloor inhospitable to fish, shellfish, and other organisms that require adequate oxygen for respiration, with consequences for both ecosystem function and commercial fisheries. The northern Gulf of Mexico hypoxic zone, which forms seasonally in waters influenced by Mississippi River outflows, carrying agricultural nutrients from a drainage basin covering approximately 40% of the continental United States, has expanded to areas exceeding 20,000 square kilometers in some years, making it one of the largest coastal hypoxic zones in the world. Similar conditions occur in the Baltic Sea, Chesapeake Bay, the Black Sea, and numerous other coastal systems, with the global area affected by hypoxia having expanded approximately fourfold since the 1950s, according to available records. Addressing nutrient pollution requires coordinated management across entire drainage basins, involving agricultural practices, wastewater treatment, and land use decisions far removed from the coastal waters where impacts are most visible. The history of scientific understanding about ocean phenomena reveals patterns of discovery that reflect both the development of observational technologies and the evolution of conceptual frameworks for interpreting observations. Many fundamental aspects of ocean function that are now taken for granted were entirely unknown, even to educated observers only 2 centuries ago, while other aspects that seemed well established have been substantially revised or overturned by more recent research. The recognition that the ocean circulates in 3 dimensions, with surface currents driven by winds and deep currents driven by density differences, developed gradually through the 19th and early 20th centuries, building on observations from naval expeditions, commercial shipping, and dedicated scientific voyages. The mechanisms driving this circulation were debated extensively, with competing theories attributing the motion of ocean waters to wind stress, temperature gradients, salinity differences, and other factors in varying combinations. The synthesis achieved in the mid-20th century, which established the modern understanding of wind-driven and thermohaline circulation as complementary components of a single global system, drew on improved observational capabilities including precise measurements of water properties at multiple depths and theoretical advances in fluid dynamics applied to rotating spheres. The development of oceanographic instrumentation has enabled progressively more detailed and comprehensive observations of ocean properties, from the earliest thermometers and salinometers used aboard research vessels to the satellite systems and autonomous platforms that now provide global coverage of surface and subsurface conditions. The Argo program, initiated in 2000, deploys a global array of approximately 4,000 profiling floats that drift at depth, periodically ascending to the surface while measuring temperature and salinity profiles and transmitting data via satellite before descending again. This network has transformed understanding of the subsurface ocean by providing continuous spatially distributed observations that were previously available only through expensive and logistically demanding research cruises. Extensions of the Argo concept now include floats equipped with biogeochemical sensors that measure oxygen, nutrients, and chlorophyll concentrations, enabling observation of biological and chemical processes alongside physical oceanography. Satellite remote sensing complements in situ observations by providing global coverage of surface properties including sea surface temperature, ocean colour related to phytoplankton concentration, sea surface height related to circulation patterns, and various other parameters that can be retrieved from electromagnetic radiation measured by orbiting instruments. The integration of observational data with numerical models has become central to contemporary oceanographic research, enabling scientists to investigate processes that cannot be observed directly, and to project future conditions under various scenarios of environmental change. Ocean general circulation models simulate the physics of ocean circulation at scales ranging from global overviews to regional details, incorporating bathymetry, atmospheric forcing, and parameterizations of processes too small to resolve explicitly. Coupled ocean-atmosphere models extend this approach to simulate the interactions between ocean and atmosphere that determine climate, while Earth system models incorporate biogeochemical cycles, ice dynamics, and other components to represent the full complexity of planetary function. These models have proved essential for understanding anthropogenic climate change because they allow investigation of how the climate system responds to changes in greenhouse gas concentrations, providing the scientific basis for projections presented in reports by the Intergovernmental Panel on Climate Change and other assessment bodies. Model development involves continuous interplay between observations, theory, and computation, with discrepancies between modelled and observed behaviour motivating refinements in model formulation and improved understanding of underlying processes. The cultural and historical significance of the ocean extends far beyond its physical and biological properties to encompass the central role that maritime activities have played in human history, shaping patterns of trade, migration, conflict, and cultural exchange throughout the world. The development of seafaring capabilities enabled human colonization of previously inaccessible landmasses, including the islands of the Pacific Ocean that were settled by Polynesian voyagers using sophisticated navigation techniques based on observations of stars, swells, wind patterns, and wildlife behavior rather than written charts or mechanical instruments. These voyages, conducted in double-hulled canoes across thousands of kilometers of open ocean, represent remarkable achievements of maritime technology and navigational skill that long predated European exploration of the same waters. The recognition of Polynesian voyaging capabilities by Western scholars came only gradually, with earlier interpretations sometimes dismissing island settlement as the result of accidental drift voyages rather than deliberate exploration and colonization. Archaeological, linguistic, and genetic evidence now documents the systematic expansion of Polynesian peoples eastward from island Southeast Asia through Melanesia, across the Central Pacific to reach Hawaii, Easter Island and New Zealand, within approximately the last 2,000 years. European oceanic voyaging, beginning with Portuguese exploration along the African coast in the 15th century and expanding to encompass global circumnavigation by the early 16th century, initiated a period of dramatically increased maritime connectivity with profound consequences for human societies worldwide. The development of navigation techniques, shipbuilding technology, and the institutional frameworks for organising long-distance voyages reflected accumulated knowledge and investment over many generations, building on Mediterranean maritime traditions while incorporating innovations suited to oceanic conditions. The Portuguese development of the caravel, a relatively small vessel combining square and lateen sails that could sail closer to the wind than previous European designs, exemplified the technological refinements that made extended ocean voyages practical. Navigation improvements, including more accurate charts, better astronomical instruments, and refined techniques for calculating position from celestial observations, reduced the uncertainties of oceanic travel. Though navigation remained challenging and imprecise by modern standards throughout the age of sail, the cumulative effect of European oceanic expansion was the creation of a genuinely global system of trade and communication connecting previously isolated regions, with consequences including the exchange of crops, livestock and diseases between hemispheres, the forced migration of millions of enslaved Africans to the Americas, and the establishment of colonial systems that shaped political and economic relationships for centuries. Maritime trade routes have historically concentrated economic activity and geopolitical attention on particular straits, channels, and ports where geographic constraints focus shipping through narrow passages or where natural harbours provide safe anchorage and convenient access to inland regions. The Strait of Malacca, connecting the Indian Ocean to the Pacific through the narrow passage between the Malay Peninsula and Sumatra, has been a focal point of maritime commerce for millennia, with archaeological evidence of trading settlements dating back to the early centuries of the Common Era, and historical records documenting the rise and fall of successive commercial empires controlling passage through the strait. Contemporary shipping traffic through Malacca ranks among the highest in the world, with approximately 1/4 of global maritime trade and significant fractions of international oil and gas shipments passing through waters less than 3 kilometers wide at the narrowest point, similar geographic choke points at the Strait of Hormuz, controlling access to the Persian Gulf, the Suez Canal connecting the Mediterranean to the Red Sea, and the Panama Canal connecting the Atlantic to the Pacific have attracted substantial commercial development, infrastructure investment, and geopolitical attention throughout modern history. The ocean's role in military affairs has shaped naval architecture, tactical doctrine, and strategic thinking from ancient times to the present, with control of the sea enabling projection of power, protection of commerce, and blockade of adversaries in ways that have frequently proved decisive in conflicts between maritime powers. The development of naval forces capable of operating effectively across oceanic distances required not only shipbuilding expertise, but also systems for provisioning, maintaining, and coordinating fleets far from home ports. The British Royal Navy's dominance during the 18th and 19th centuries rested on a combination of quantitative superiority in ships and personnel, qualitative advantages in seamanship and gunnery developed through extensive experience, and institutional capacity to sustain global operations through a network of bases, dockyards, and supply arrangements spanning the world's oceans. This naval power served commercial interests by protecting British merchant shipping while threatening that of competitors, and it served imperial interests by enabling the projection of military force to distant possessions and the maintenance of communications with far-flung colonies. The transition from sail to steam propulsion during the 19th century and subsequently to nuclear power in the 20th century transformed naval capabilities while raising new requirements for fuel supply, maintenance infrastructure, and technical expertise that influenced both military strategy and economic development. The contemporary ocean economy encompasses activities ranging from traditional fishing and shipping to emerging sectors such as offshore renewable energy and marine biotechnology, contributing trillions of dollars annually to global economic output while employing tens of millions of people in ocean-related occupations. Shipping remains fundamental to international trade, with approximately 90% of global merchandise trade measured by volume moving by sea, carried by a fleet of commercial vessels ranging from small coastal traders to enormous container ships and bulk carriers capable of transporting hundreds of thousands of tons of cargo. The containerization of maritime trade, which developed from the 1950s onward and became dominant by the 1970s, revolutionize shipping efficiency by standardizing cargo handling and enabling rapid transfer between ships, trucks, and trains without unpacking and repacking goods. This transformation reduced freight costs dramatically, contributing to the globalization of manufacturing by making it economically viable to produce components and finished goods at geographically dispersed locations and assemble them through complex international supply chains. Offshore energy production has expanded substantially over recent decades, with oil and gas extraction from continental shelf waters providing a significant fraction of global hydrocarbon supply, while offshore wind installations have grown from negligible capacity to a substantial component of renewable energy portfolios in several countries. The technical challenges of operating in the marine environment have driven innovation in engineering, materials science, and remote operations, creating capabilities that extend from shallow coastal waters to depths exceeding 3 kilometers in the case of some oil and gas developments. Offshore wind farms, initially installed in the relatively calm and shallow waters of the North Sea and Baltic Sea, have expanded to more challenging environments as technology has improved and as experience has accumulated with the installation, operation and maintenance of marine wind turbines. Floating wind platforms, which can be deployed in deeper waters where fixed foundations are impractical, represent a recent technological frontier with pilot projects demonstrating feasibility, while commercial-scale developments remain in early stages. The potential for wave and tidal energy conversion has attracted research investment and prototype testing, though these technologies have generally proved more difficult to commercialize than wind power due to the harsh operating environment and the challenges of transmitting power from marine installations to onshore grids. Marine biotechnology represents a growing area of economic activity and scientific research, seeking to develop commercial applications from the enormous biological diversity of marine organisms and the novel compounds they produce. Marine organisms have evolved biochemical adaptations to extreme conditions of pressure, temperature, salinity, and radiation that differ substantially from those encountered by terrestrial organisms, creating potential sources of enzymes, pharmaceuticals, and other valuable compounds with properties not found in terrestrial equivalents. The thermostable enzymes isolated from organisms living at hydrothermal vents have found applications in molecular biology and industrial processes that require functioning at elevated temperatures, exemplifying the commercial potential of marine biological resources. Drug discovery programs have screened marine organisms for bioactive compounds with potential pharmaceutical applications, identifying candidates for treating cancer, infectious diseases, and inflammatory conditions, though the path from promising screening results to approved therapeutics typically requires many years of development and faces high failure rates. The governance of access to marine genetic resources and the equitable distribution of benefits derived from their exploitation has been a subject of international negotiation, with recent agreements under the Convention on Biological Diversity and the United Nations Law of the Sea framework, seeking to establish principles for benefit sharing that recognize both the contributions of countries in whose waters resources are found and the investments made by researchers and companies in developing commercial applications. Tourism and recreation centered on the ocean constitute another significant economic sector, with coastal destinations attracting visitors for beach holidays, water sports, diving, whale watching, and other activities that generate substantial revenues for local and national economies. The aesthetic and recreational value of marine environments creates economic incentives for conservation that complement ecological and ethical arguments for protecting ocean ecosystems, though these values can also conflict with conservation objectives when tourism development damages the ecosystems that attract visitors. Coral reef tourism provides an instructive example, with diving and snorkeling on reefs generating billions of dollars annually for countries such as Australia, Indonesia, and those in the Caribbean, while simultaneously creating impacts from anchor damage, diver contact, and infrastructure development that can degrade reef health. The management of tourism to maximize economic benefits while minimizing environmental damage requires understanding of both ecological sensitivities and visitor behavior, along with institutional capacity to implement and enforce appropriate regulations. The study of the ocean continues to advance through new observational capabilities, improved modelling approaches, and innovative conceptual frameworks that reveal previously unsuspected connections and processes. The recognition of microbial dominance in marine biogeochemical cycles, for example, has transformed understanding of how the ocean processes carbon, nitrogen, and other elements, revealing that bacteria and archaea conduct the majority of chemical transformations that determine ocean chemistry and atmospheric composition. Advances in genomic sequencing have enabled characterization of marine microbial communities at unprecedented resolution, revealing extraordinary diversity and identifying metabolic capabilities that had not been anticipated from traditional cultivation-based approaches. These findings have implications for understanding the ocean's response to environmental change, because microorganisms can adapt rapidly to changing conditions through both physiological adjustment and evolutionary change, potentially buffering or amplifying the effects of perturbations on biogeochemical cycles. The interdependence of human societies and the ocean becomes increasingly evident as scientific understanding deepens and as anthropogenic pressures on marine systems intensify. Climate change, ocean acidification, overfishing, pollution, and habitat destruction collectively pose challenges to ocean ecosystems that may exceed anything in human experience, while simultaneously threatening the services that healthy oceans provide to human societies. The responses to these challenges involve scientific research to understand processes and project future conditions, technological innovation to reduce impacts and develop sustainable alternatives, policy development to regulate activities and protect vulnerable ecosystems, and social and economic adjustments to enable transitions toward more sustainable relationships with the marine environment. The outcomes of these efforts remain uncertain, depending on choices yet to be made by individuals, organizations, and governments, whose decisions will shape both human welfare and the condition of ocean ecosystems for generations to come. The ocean that future generations inherit will reflect the cumulative consequences of countless decisions made under conditions of incomplete knowledge and competing priorities, continuing a pattern of human-ocean interaction that extends back to the earliest coastal settlements and forward into whatever futures these decisions create.